In this video, we are going to watch another video regarding polymerase chain reaction. Before I go and watch this video together, I am asking you to do this part by yourself in the future. After watching this video, then you can go to your PowerPoint file and then go to click on this link and try to do some search on this website and get some information by yourself. It has a lot of information regarding DNA, about DNA techniques, about the amplification, and other things. So you need to search and find out by yourself as your task. So right now I am going to go to this website and click on that and watch this video together. Okay, if you click on that link, then you have to download, and it is a kind of SWF file. So SWF file, and you need to have an application for opening that file. Okay, let's listen together.
Any copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
Okay, we get back again to the PowerPoint file. To summarize what is PCR, it's the way to produce a large amount of the DNA, and it has 3 major parts or steps. First, denaturation, which means at this stage the DNA sample should be heated until 95 degrees centigrade in order to get separated, or in order to have 2 complete separate strands of DNA. And then the next step is annealing, which is the step for adding primers. You can see the primer attachment over here. So primers are attaching to both strands of DNA from 2 opposite directions. And then the third step is elongation, in which DNA polymerase start to add polynucleotides. And the adding polynucleotides is in the direction of 5' to 3', which is like from here, and again from here, again 5' to 3'. This is how DNA polymerase is adding nucleotide to each DNA strand. And you already know that how the number of the copies will be calculated using this formula. If there is a 30 cycle for this PCR, then at the end 2 power n, which means if it is 30 cycle, then that would be 2 power 30. Then you will have 1 billion, more than 1 billion copies of DNA after 30 cycles. Do you have any idea how long does it take 30 cycles in a PCR? Pause the video and think. Okay, you answered correctly, almost. It takes about something like 2 hours. So in 2 hours, you will have 1 billion copies of DNA. Isn't it interesting and amazing? Yes, it is. Okay, the next topic that we are going to talk is about the theories or what scientists thought about production of the DNA as 3 models, and how Meselson and Stahl, as 2 scientists, proved that the model is correct. So first of all, let's see what are these models. The model which was offered by Meselson and Stahl is called semi-conservative. I think you almost know about this model. We already talked about that. The other 2 models is conservative and dispersive. Let's see what does that mean if it was going to be a conservative model for DNA amplification. In the conservative model, first of all, do you know what's the meaning of conservative? Yes, means conserving or keeping the first DNA and completely making a new one. So in this model, you will see that the first DNA as a DNA with 2 strands will be conserved or kept completely as original, and then a complete new DNA will be produced. So in this model, the original DNA completely conserved, and the new DNA is completely new for both strands. That's the reason it is called conservative. In the model of dispersive, it's kind of dispersion or spread out all the types of all the parts of the DNA becoming a new one while they are replicating or while they are amplifying. It's a little weird, I think, dispersive model, but anyway, that was another another theory that was offered by these at that time. For now, we are going to prove why the Meselson-Stahl model is correct rather than the other 2 models. In order to do that, first of all, we need to understand what exactly they did in their experiment. So I explain a little about their experiment over here, and then I show you the video we watch together, and then I try to draw whatever is happening during this Meselson and Stahl experiment in a new note, and you will notice exactly what's going on. Okay, so the first step is that the experiment started with a nitrogen medium including nitrogen 15. So in the first airline, there are bacteria which are growing, and the medium includes nitrogen 15, which is a heavy radioisotope from nitrogen. And if they are going to spin using centrifuge, the result would be the only bacterial culture going down the centrifuge in each tube because it has only nitrogen 15 as a heavy radioisotope. The next experiment is that then they transferred the bacteria to a culture in which the culture had nitrogen 14, which was a lighter nitrogen. What happened in that case, after centrifuge, they found a middle band, not a heavy band. So if you want to divide the bands observed, the first band is observed here as a heavy one, and then at this experiment the second band, the next band in this experiment was observed at the middle, and then again they cultured for the next generation. This is the first generation and this is the second generation, and in the second generation they observed the middle one and they found a lighter band. They didn't see any more the heavy band. They didn't see any more the heavy band. Pause the video and think about a little about that. Can you say why? Okay, thank you. I think you already understood. However, I'm going to explain it completely again. So, first generation, second generation, and then the third generation. Again, in the third generation, only the middle band and the lighter band. This is what they observed, and then they started to analyze what exactly happened and why this heavy band was removed and it wasn't observed in the first generation or the second generation or the third generation. No heavy band in none of them. Okay, let's watch the video of Meselson and Stahl experiment together.
The Meselson and Stahl experiment provides evidence for semi-conservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Meselson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
In order to understand what happened during the Meselson and Stahl, here I have already drawn a picture from the first tube, and also there is a question regarding the Meselson and Stahl experiment. I'm going to ask you to read this question and answer this after my explanation in the next video. Okay, so in the first centrifuge result, they found only a heavy band which is located over here. So let's do analysis and draw the DNA together. If this is the DNA band having the nitrogen 15, then I am drawing it with the red color. And then let's do for the first generation and see what happens in the first generation. So in the first generation, the two DNA strands are separated, you see with the red colors. And in this first generation, if you remember, the bacterial culture were moved to nitrogen 14. So the new strands includes nitrogen 14, which I am showing with the blue color, not red color. So what happens? All the bands, all the DNA generated, okay, all the DNA generated includes the red and blue color, red and blue. So this DNA and all the replication or amplified DNA, they have red and blue color, or they don't have both of the red, which is showing the heavy band. So in this case, it is slightly lighter. How much lighter? Is exactly the half light, because half is heavy and half is light. So it sits in the middle part after centrifugation. This was the first generation. And then what happens in the second generation? You just imagine this DNA is separated, red is here and blue here, and each of the strand will get a new strand. Because all the culture includes only nitrogen 14, you will see that always with the blue color because it shows the nitrogen 14. So this DNA has red and blue, but this one, which is coming from the replication of this strand, it has only blue color, which means nitrogen 14. And again, this one, it has only blue coming from this strand, or amplified from this strand, and red blue. So the production or the results of the second generation is including the DNAs having red and blue, which is locating in the middle, and the DNA is only blue. DNA is only blue is including only nitrogen 14, and nitrogen 14 is lighter, so it sits over here. So nitrogen 14 is here, and here is nitrogen 14 plus 15, and here, which is both red, which is not observed over here, only nitrogen 15. So with this, we continuing with this model, then you can draw the third generation. The third generation, again, separation of the DNA strands, and then making a new one with the blue color showing the nitrogen 14. So each strand I have already drawn like this. Each strand can create two other strands. Half is this and half is this. The blue one can create only blue, and the red blue can create half red blue and half blue. Again, at the third generation, you will see only a few of these products have the half blue, half red. Only a few of them, and most of them have blue. So we will have again a middle band including nitrogen 14 and 15, and the upper band only nitrogen 14. And if you continue for the fourth generation, you get the same pattern. Again, the same pattern with a blue, red, and blue, blue. Okay, after this explanation, you are supposed to be able to answer this question. So please pause the video and read the question and answer all of them. And I'm going to answer these in the next video. Okay, thank you.